Hello, everyone. Welcome to the new session of a chapter called Biological Classification. This is the fourth session of this chapter, which deals about fungi. So, fungi got its position in Whitaker Five Kingdom classification. Okay, and it is a large kingdom which is having more than 100,000 species reported. Fungi are achlorophyllous. Achlorophyllous means they don't have any chlorophyll. So they cannot make their own food. So that is why they are heterotrophic mode of nutrition they follow. They also produce spores. Generally, they reproduce by producing spores. And there are different types of spores. We will see in the next slides. They are non-vascular in nature. Non-vascular in nature means they do not have any water conducting tissue. As we see in the case of pteridophyte, gymnosperm and angiosperm, they are having very specific water conducting tissue. So that is totally absent in the case of fungi. They are eukaryotic organism. Okay, so they are having well developed nucleus in its cell, and it is also having plasma membrane and surrounded by a cell wall. But its cell wall is different from the plant cell. How it is different? The plant cell wall is made up of cellulose, whereas fungi cells having a cell wall made up of chitin. That is a type of carbohydrate only, but is different from the cellulose. Okay, so the study of fungi is called as mycology. If I am studying fungi, then I'm studying mycology. So this is very, very important. Study of fungi is called mycology. Okay, they are absorptive type of nutrition. What does this mean? Absorptive type of nutrition. Absorptive type of nutrition means the cell release some chemical. Generally, if it is, I'm drawing a cell. Suppose this is a cell. It's having a nucleus. So what does, and it is growing on a dead decaying matter. So this is a dead decaying matter. Okay, so it releases some chemicals. And those chemicals may in the form of some enzymes. So what does this enzymes or the chemicals do? It digest or make the complex substance available in the dead decaying matter into a simpler forms, and it absorb it. When it get changed into simple substances, okay, complex substances are changed into simpler substances, then it get absorbed inside the cell. So they are either parasitic or saprotrophs. So generally the fungi are parasitic or saprotrophs. So this is absorptive type of nutrition. So in both the cases, like in parasites and saprotrophs, absorptive type of nutrition is followed. Okay, so now let us study about some of the characteristics of fungi. So fungi constitute saprophytes and parasites. Saprophytes means they are responsible for the spoilage of food and act as the decomposers and mineralizers. Means, so these fungi grow on a dead decaying matter. Okay, and they take the food from the dead decaying matter. How does it take? As we have discussed, they are having absorptive type of mode of nutrition. So they release out chemicals or enzymes on the dead decaying matter, and what they do, it changes the complex food substances into simpler form. So those simpler food substances is absorbed by the fungi. So they are also called as decomposers. Now here it is written, they are decomposers and mineralizers. So why they are called decomposers? Because they decompose the dead decaying matter. Okay, and mineralizers, because in these dead decaying matter lots of minerals are present, so it mineralizes means minerals are released from the decomposed matter, and those minerals are available in the environment, as well as some important minerals which are needed by the fungi, it absorbs them. Okay, so they are also called as decomposers or mineralizers. Now, the parasites cause various diseases in animals, man, plants, and results great loss. So, the fungi are having a causing great loss to the animal, man, plant, and okay. So, animal, man, and plant, they causes great loss to them. How? Let us see. So they are parasites, and they are of two types of parasites. If they are present on the body of organism, whether it is a plants or animals, it is called as ectoparasite. And when it is present inside the body, then it is called as endoparasite. And it causes various types of diseases in plants and animals. For example, white spot disease and mustard. This is very important for if we talk about the examination point of view. White spot diseases in mustard plant, and it is caused by fungus albugo. Okay, white rust disease caused by puccinia. Sorry, wheat rust disease caused by puccinia. So this is also one of the very important disease caused by fungi, puccinia. And it has been asked in many times in the examination also. Okay, saprophytes are commonly seen in the decaying and dead organic matter. So they act as a saprophytes also. What are some of the saprophytes? Yeast, rhizopus, mucor, agaricus. These are the some of the examples of fungi which acts as a saprophytes. Some fungi are caprophilous, means they also grow on the fecal matter of animals. So that is why it is called as coprophilous. So coprophilous fungi. Symbiotic forms are seen in association with the algae and lichens. Now, this is very, very important. Algae as lichens. So, what happened in lichens? There is again symbiotic relationship. Symbiotic relationship means two organisms comes together, okay, and they live together, and it act as a one organism. So here the two components are there. One is fungi and one is algae. So what does the algae do? Algae do have chlorophyll in it, and it absorb all the sunlight and do the process of photosynthesis. Whereas fungi, what does it do? It absorbs water from the substrate and it provide to the algal cells. So in the process of photosynthesis, water is required. So water is made available to the algae with the help of fungi. Also, fungi form a mesh-like structure in which algal cells are protected. So this type of relationship is called as lichens. Okay, so so they can be in a parasite mood, they can be as a saprophytes, and they can also found in a symbiont form in a form of lichens. The body of fungus is called mycelium. Okay, so if we talk about the body of fungi, then they are a thread-like structure like this. Thread-like structure. This is a single thread-like structure I'm drawing here, which is called as hyphae. This is hyphae. This is hyphae. Now there are numerous thread-like structure like this. And this is called mycelium. So this is the body of the fungus. Mycelium is called as the body of the fungus. Mycelium generally consists of thread or a tube-like cylindrical filamentous structure which is called as hyphae. So here we have seen hyphae is a single thread-like structure, and when numerous thread-like structure together they form a body of the fungi, then it is called as mycelium. Hypha has a thin wall with the inner lining of cytoplasm. So if we see a single cell of hypha, it is a eukaryotic cell. So cytoplasm is there, plasma membrane is there, okay, and they also have a cell wall. But here the cell wall is non-cellulosic, means it is not made up of cellulose. Instead, it is made up of chitin, which is another polysaccharide. Okay. Hyphae with a septa is called septate, and without septa is known as aseptate. So here we have seen the hyphae is, if it is septate, septate means like this: 1 septa, 2 septa, 3 septa, 4 septa. Okay, so this is septate hyphae, but sometimes the hyphae is aseptate, means nucleus are present like this. So numerous nucleus are present like this. So sometime the hyphae are septate and in sometime they are aseptate. Septate form are uninucleate, but aseptate are syncytic. So I have already drawn the a diagram of a septate hyphae. Again, I am drawing it. Septate, this is 1 cell, this is 2 cell. So single nucleus is there. Okay, single nucleus. And when there is aseptate hyphae, numerous nucleus is there. So when numerous nucleus is present in the cytoplasm, it is called as syncytic hyphae. And when it is septate, they contain single nucleus in it. Single nucleus in it. Okay. Oil and glycogen are the storage form of food. Okay. So when food is stored in fungi, it is stored in the form of oil and glycogen. Okay. Special vesicles having wall material occur near the hyphal tips. So at the hyphal tips, they are having special vesicles which is called as chytosomes. Chytosomes. Okay. Reproduction. So if we talk about the reproduction in fungi, there are 3 different types of reproduction reported in case of fungi. It is in vegetative form, it is in asexual form, and it is also reported in sexual form. If we talk about the first one, that is the vegetative reproduction, it follows as fragmentation, fission, and budding. 3 different types of vegetative reproduction are there: fragmentation, fission, and budding. So fragmentation, the mycelium breaks into several fragments and each fragment later develops into the new fungal body. So suppose there is a mycelium, long thread-like structure, hypha is there, okay, and when it breaks down, so we have 2 different hypha like this. So each hypha or each like this mycelium, okay, fragment can grow a new fragments, a new thread-like structure and form a unit body of a fungi. So like this, fragmentation helps in reproduction. Next is fission. This kind of multiplication is seen in unicellular forms of fungus like yeast. So generally, yeast, what does it do in this type of reproduction? Suppose this is a yeast cell. Okay, so what happened at the time of fission, like you have seen in the case of amoeba? Amoeba do its fission in a binary fission form. So in the next stage, what it do? It follows by the karyokinesis, in which the nucleus constrict. So as you have seen in the case of amoeba also, nucleus of cell get constricted and it divide into two nucleus. In later stage, the nucleus divide into two nucleus. Now the cytoplasm also divide. Next, what happened? This cytoplasm also divide into two form, and each nucleus gets surrounded by a bit of cytoplasm. Okay, and two daughter cells are formed. So this is a type of a fission. Nucleus undergo constriction followed by its division, and further the division of cytoplasm and cell wall results into a two daughter cells. So in this way, yeast cell undergo the process of fission. Budding. So this is a very common type of vegetative reproduction found in yeast. So what happens in the case of at the time of budding? A small outgrowth is formed from any side of the yeast cell. Nucleus goes to that particular site and a small outgrowth is given. So nucleus also divide at that particular tip point where the bud is formed. As you can see here in the diagram, this is the bud part. This is the bud part, and the nucleus is also dividing at this particular part. So one nucleus goes to the bud and another is present in the parent cell only. Later, this bud is cut off from the parent cell and a new cell is formed. But here, if you see the difference between the fission and budding, there in fission, the cells are of equal size. The nucleus divide at the center. Here, the nucleus is not dividing at the center of the cell. Okay, it is dividing at the periphery where the bud is formed. So, this is the difference between the fission and budding. And also when this bud is sometime get pinch off, but it is still attached to the parent cell. And later you can see this, the first bud also undergo a division by forming a new bud. And this again also form a new bud. And this again a form new bud like this. And you can see a chain of bud in this way. Okay, so the bud breaks off from the parent cell and develops into a new individual. And we can see a bud of chain in this form. So this is a very common type of vegetative reproduction reported in the case of yeast. Next is asexual reproduction. So this is also very common in case of fungi. So the common method of asexual reproduction is by spore formation. So there are different types of spores formed in fungi. We will see what are those types. The spores may be exogenous, external, or endogenous, internal. Now this is a very important point. Exogenous, that is external, it is formed external to the body, and endogenous, it is formed internal to the body. The externally formed spores are the conidiospore. This is also very important. Sometimes it is asked in the examination. And the internal producer spores are known as sporangiospores or zoospores. So how the conidia is formed? Conidia are formed at the conidiophore. A stalk-like structure bears conidia. This is the conidia. So this is conidiophore and this is conidia. This is conidia. The spore is called conidia. And this stalk-like structure is called as conidiophores. And these spores are called as conidiospores or conidia. And internally, now here if you see in the diagram, sporangium is there. This is called sporangium, a stalk-like structure which is called as sporangiophore. And in the sporangium, nucleus divide and it is surrounded by a bit of cytoplasm. Later, the cell wall is laid around this bit of cytoplasm and it forms spores. When this sporangium is dehisces, means it ruptures, it releases lots of spores out from this sporangium. So this is the dehisced sporangium and the spores are released here. So this, it is formed inside the body of sporangium, whereas conidiospores or conidia, it is formed outside the body. It is formed outside the body. So that is why it is called as externally formed spores. Whereas sporangiospores or spores or zoospores, it is formed outside the, it is formed inside the body and later it is released outside the body. Conidiospores are the non-motile spores formed in a chain by the constriction and separation of fungal hyphal tips. So here you can see a chain of conidia. So this is conidiophore. This is stalk-like structure on which conidia are born is called as conidiophore. So this is in case of penicillium, conidiophore. And on the, if you see on the conidia, there are chain-like structure. This is the chain-like structure. Here again, you can see this chain-like structure. So the oldest conidiospore is at the tip. Oldest conidiospores is at the tip, whereas the new one is at the base. This is a very important point to remember because as we have seen in the case of budding in yeast, okay, in the same way the conidia are formed, but they are not separated and they are attached. Okay, so in this way it forms a chain of conidia or conidiospores. Now here sporangiospores are the non-motile and zoospores are the motile and formed inside the hypha. So here we can see the sporangium. Okay, so some of the aquatic fungi like saprolegnia, they release zoospores. Zoospores means they are having flagella and it can swim in water. So that is called as zoospores. But the sporangiospore, these sporangiospores, these are non-motile. They cannot move from one place to another. Okay, so they are generally a powder like structure, small powdery mass like structure, and they are released from the sporangium. So they are non-motile, whereas some spores which are having some flagella like structure, they are called as zoospores. Sexual reproduction. The sexual reproducing fungi may be homothallic or heterothallic. Now this is very, very important. Homothallic or heterothallic. So sometime the fungi are having hypha Okay, and these hypha can come in contact together. And what happened? The hyphal cell fuses and their nucleus get mixed. As we know that in the sexual reproduction, the male gamete and female gamete fuses together. Okay, so here also this the hyphal cells, okay, having nucleus, they come together, the two hyphal cells fuses together of a different hyphae, and their nucleus get fuses together to form zygote like structure. Now, homothelic means the thalli or the hyphae are of same type. Heterothelic means the hyphae are of different types. So sometimes their hyphae are noted down as plus strain and sometimes it is noted down as minus strain. Plus minus means there is no demarcation as a male hypha or a female hypha, but the strains are different in the case of fungi. So it is denoted as plus and minus strain to just differentiate the different types of hypha. Okay, so if it is plus and minus strain, then it is called as heterothelic. Male and female sex organs are formed in the same thallus in homothelic. So here the male and female sex organs are formed in the same thallus, and that is why it is called as homothelic. Whereas these are formed in the different thallus. Okay, thalli are different, then it is called as heterothelic. Sexual reproduction in fungi take place by somatogamy. As I have told you here, that the fusion of two somatic hypha acting as a gamete. They are acting as a gamete and it is called as somatogamy. Now, the fusion of the cell can be of three different types. It can be isogamy, it can be anisogamy, and it can be oogamy. Now, what is isogamy? Isogamy, the gametes are morphologically and physiologically similar. You cannot differentiate that this one is a larger or this one is a smaller. Okay, morphologically and physiologically both are similar. So that is why both are isogamous type and their union is called isogamy. Anisogamy. As you can see here, one is larger and one is smaller. One of the gamete is larger in size and one is smaller in size. So anisogamous, morphologically or physiologically, they are not similar. So anisogamous type of fusion. Now oogamy. In oogamy, the egg is formed and this egg is female reproductive organ. It is non-motile. Generally, it is non-motile. And sperms that are motile in nature. So this is oogamy. Sexual reproduction results by the fusion between the similar types of gametes. Again, it is about isogamy when reproduction takes place by the fusion between similar types of gametes. Okay, the fusing gametes are exactly similar in appearance and function. Morphologically and physiologically, they are quite similar. Anisogamy. The fusing gametes are of different in either physiological or morphological features. So they are morphologically or physiologically dissimilar in nature. If the fusing gametes are of morphologically similar and physiologically different, okay, morphologically similar but physiologically different, it is known as physiological anisogamy. Now here two types of anisogamy: physiological anisogamy. Morphologically the gametes are similar but physiologically they are different. So that is Rhizopus and Mucor. Such type of anisogamy is reported in Rhizopus and Mucor. Now, if the fungus gametes are different in both physiological and morphological feature, okay, so they are differentiated, they can be differentiated in both the ways, physiologically as well as morphologically. So it is known as morphological and isogamy. Now here, this is a figure of oogamy in the case of fungi, fusion of male gametes, and it is called as enterozoids because they are having flagella. Okay, and entero means anther-like, means male reproductive structure with a large and non-motile female gametes. Here the egg is quite larger, okay, and it is non-motile. So union of or the fusion of this male and female gamete, it is called as oogamy. It shows male enthedial and female oogonial development. The former produces, example, it is in case of pythium. Fusion between two hyphal gametangia produces egg and sperm. Sometime take place, and it is called as conjugation in the case of Rhizopus and Mucor. Okay, so there are two different types of, we can say, the oogamy. First, in the case, it is male, and that is enterozoids and egg development is there. And in the second case, the hyphal gametangia produces egg and sperm. Now, this hyphae is quite different from the somatic hyphae. Okay, so this is called, that is why it is called as hyphal gametangia, and it produces egg and sperm. Okay, so this is reported in the case of Rhizopus and Mucor. Classification of fungi. Now, fungi are classified on the basis of morphology of vegetative mycelium, morphological features of a reproductive structures, types of spores and its dispersal, life cycle, physiology, and biochemistry. So, on the basis of these characteristics, okay, there are 4 different classes of fungi. They are namely phycomycetes. It is also called as algal fungi. Ascomycetes, sac-like fungi or sac fungi. Basidiomycetes, club fungi. And deuteromycetes, imperfect fungi. So the above 3 classes, they have reported sexual reproduction, but in the case of this deuteromycetes, that is imperfect fungi. Why it is called imperfect fungi? Because the sexual reproduction has not been reported, and the fungi which is found or invented a new fungi, and there the sexual reproduction is not reported, okay, till date, it is kept in deuteromycetes. And later on, when the sexual reproduction is reported or found and or can be seen by the scientist, and suppose it is a type of ascomycetes, then that fungi could be kept in ascomycetes. But till date, if the fungi has not been reported its sexual phase, then it is kept in deuteromycetes. So here we are having 4 different classes of fungi. In the next class, we will study about the all 4 different types of fungi in the new session. Okay, so thank you very much for this session. We'll meet again with the fungi and virus part of the biological classification in the next session. Thank you very much.